I will start with a very general introduction about parasite in aquatic food web, and then going quite directly into the kitchen work. I'm also a kitchen person, so this talk will focus pretty much on chytrid. So we will have a bit closer look on the microbial loop, and then a bit of overview on different approach and method that we can use to study a bit more what are the mechanism and the processes behind the microbial loop, and how zoosporic parasite can support trophic transfer in aquatic environment. So I prepared the talk. If I'm getting a bit too long, just let me know and we can stop, and then I will share the slide and then I'll do the interpretation of the end. Anyway, so yeah, just very short. Which is the arrow for going?
It's not working.
I can try with the mouse. Oh yeah, I can use the mouse. Okay, so just a first slide has to say a very general overview on how we will look at parasite during this talk. Well, we know parasite have very important ecological roles. Among them, the regulation of host population, which is a kind of important topic for the action. But yeah, what we will look at today is how directly contribute to energy flow in food web. So, parasitism is first of all a trophic strategy. So parasites are consumers, and as we know, it's a bit different from predation. They are not really prey-side limitation and no trophic level limitation. So parasite can almost target every organism. And also what we will have a very close look at is how parasites get consumed, either with the infected host, but also as a free living stage, as a food source for other organisms. So yeah, this was just to mention quickly that yesterday Kevin explained us quite good. So my topic was intended to be before, and so I just wanted to briefly mention that parasites are important on this point of view as a consumer, and they are consumed, but they are also important in the structure of the food web. So including parasite add trophic link, and this is also another example of a food web, how parasite can include, first of all, if we take into account parasite in the food web, we include species, we increase the number of taxa, so of course we increase the number of links, but this is not really a direct relationship. Adding parasite increase by threefold was estimated in this work, the trophic links, because as I said, parasites, they are not only consumers, they only get consumed, so it adds quite a lot to complexity. And what I want to add also with this slide, these studies that has been conducted now quite a few years ago, is that at the time it was not estimated the number of parasitic protozoa, viruses, and fungi and bacteria. So this is something that we could get in with also using new technology, molecular ecology, and so it's something that is still in progress and adding knowledge to our conception of food webs. So this is a very recent work. There was last year quite interesting special issue in Ecologia. And so the author estimated the link in a lake. So they separated the link, they estimated the benthic, the pelagic, and the total lake compartment. And this is a bit to show you quantitatively what I was telling before. So the blue bar is the predatory parasitic link, so the link that involve parasite has a trophic source, and the green one is the purely parasitic link. So when we take into account a total lake food web, we see that the links with the predator, the prey and predator links, and the parasitic predator link are kind of equivalent. But moving to the pelagic compartment, it's getting quite important. Also, this number of link of predation on parasite. And well, I'm just saying that because I focus also a lot on pelagic compartment. So we will have a look during this talk mainly on what are microbial and pelagic links in aquatic environment. So just to have a bit of focus now, what are the parasite trophic link that we will tackle in this talk. So as we had very good information yesterday, parasites are a polyphyletic group, so they encompass different organisms. They can be endobiotic or epibiotic, so endoparasite or ectoparasite. The thing is that, as we say, they produce motile spores as infected stage, so they are particularly adapted to the aquatic lifestyle. And well, here a bit of overview, let's say simplified overview of what can be the target of this parasite in aquatic environment. So as we say, we have basically all the food web level from the phytoplankton to fishes. And so today we will focus mainly on zoosporic parasite of phytoplankton that produce these spores. Yesterday Alena showed me very nicely in a picture, produce this globule oil. So zoospores do not feed, they have their own reserve. And so this is what we will focus on. And also on the link with these two compartments, the micro and meso zooplanters. So I just wanted to add also my personal homage to, well, Sparrow and Hidakante. Well, in the picture that I took before from a work from Frank Gleason, well, he could represent quite nicely a bit of overview of zoospore. But this was how they were described already in the early 60s. And well, Frederick Sparrow did quite a lot of work in describing all the aquatic phycomy set, while Hinda Canton really was focusing on chytrids. So these are quite historical reference. I think now it's quite easy also to find it on the net. I don't know, I remember at the time of my PhD, I could buy some second-hand book. So that's the way how I got the Sparrow and the reference from Hilda Cantor. So, I mean, I hope that now there are more accessible, but there are ways also to find this reference. And well, as you can see, the representation is really quite amazing. Anyway, so, well, this just to, I wanted to make a precision with this slide. So as I say, well, the chytrids have these two sides. So of course, on one side, we study them as a parasite, the infection dynamic, their effect on the host. And today we will focus on the food web energetic and trophic ecology. What I want to say, yesterday was Kevin talk, and Kevin explained us quite good that while feeding on a dissemination state of parasite is a strategy for parasite to infect secondary, tertiary host. So it's a way of, closing the cycle. While chytrids have a very simple life cycle, they only infect, the chytrids that we will talk about, they only infect algae, they do not have intermediate host. So when a consumer feed on zoospore, it's really a nutritional resource. I mean, we have no evidence that feeding on chytrid zoospore is a way of transmitting the the infection. So I just want to make this precision to, precise the point of view. So we will really have a look on what means feeding on the zoospore has nutritional results. This has nothing to do with the chytrids life cycle or the spreading of the infection. Okay, so let's dive in our pelagic food web and the trophic links. So when we think about a pelagic food web, since now a few decades, we know very good that most of the links are microbial. It was the microbial loop that was really focusing a bit on the importance of the heterotrophic microeukaryote as key compartment to transfer the energy from the very basal level, the small phytoplankton, the bacteria, up to filtration zooplankton and the upper trophic level. But so this to say that, for kind of some time, the pelagic food web links really focus on, typically predation, herbivory. So this was, something that was quite strong in the conception of these food webs. And the large phytoplankton, besides contributing to the solid organic matter pool, were mostly considered a sort of sink link, in the sense that a lot of the organic matter that was in the large phytoplankton was lost by sinking, because large phytoplankton is not really grazed by the consumer. So the size constraint were making this link not happening. And so it was, it was Maiko, already a few years ago now, that conceptualized the mycoloop. She was working on Lake Biwa, on diatom and desmid, and she observed that, algal infection preferentially affect large algae. And so, as we said, these large algae are not directly exploitable as nutritional resources, but so she understood that, the fungal infection and the production of zoospore could be this alternative link to the consumer. And so the conceptualization of the microbial loop, the mycoloop, opened the way also for studying in deep these dynamics and these links. When I started studying chytrids, I thought, okay, but I want to study a new trophic link. I want to see if there can be also some meaning of this parasite for, cyanobacteria, toxic bloom. So I wanted to see if this concept could be a bit extended also not only to diatom and desmid, but also to other phytoplankton, large or filamentous species. And so from there, we start moving and thinking a bit about, how to say, a bit more in-deep questions. So, can chytrids support the consumers via the production of zoospore? So are zoospore enough to support these consumers? And so if chytrids are an alternative link, how this link can work. So are chytrids improving the nutritional quality of the resources from different algae, and what are the importance? So what are the underlying mechanisms, and what is the importance in the current world? So considering the current scenario, actually is, we all know the global warming, the increase in extreme weather events, and since quite a long time now, we know that there is a direct link of this change with the proliferation of harmful toxin bloom. And this bloom often considered what are called the trophic dead hands. So they are considered that, yeah, they are not exploitable by consumer. And so the question were, okay, so can we frame our knowledge about mycoloop and chytrids within this bit more larger concept? So I will go through a few approaches that I used and colleagues also used. So I will start a bit with some data integration and modeling. So when I finished my PhD, we thought, okay, it can be nice to have a bit general view, but we missed some measures. Some of this flow were not measured directly. So inverse model are based on mass balance. So it can be a nice way to try to calculate this flow that were not measured directly. And then, of course, we moved from modeling a bit more to, well, let's say the real world, so, or at least more empirical evidence with the lab experiment, so control measure of grazing and host growth. And here I will cite the work from colleagues, while mainly Thais was also doing a lot of work with different models. And then I will say getting a bit into detail with some information about, well, what we did on the biochemical profile and nutritional quality of this organism. Okay, so let's start with the inverse model analysis. So during my PhD, well, I studied eutrophic lake, but nevertheless, we also studied oligomesotrophic lake, where we observe a bloom of diatom during spring that was also heavily infected by parasite. And so what we did, we start well, modeling the carbon flow without chytrid, and then we wanted to compare. So these are, well, the focus, we will have focus on this compartment. So the large phytoplankton, so phytoplankton on the size range between 20 and 150 micron. The detritus pool, the microzooplankton and the mesozooplankton. So what we observe is that when there is a bloom of large phytoplankton, indeed, there is quite a lot of this organic matter that directly is lost by sinking, and also it's contributing to the detritus pool and then sinking again. So what we did, we had the flow that, well, we knew was calculated from the parasitic infection. So, well, what we estimated, it was the flow that was conveyed from the large phytoplankton to the sporangia, and then from the sporangia to the zoospore, and then to the consumer. So here what I want to precise is that, well, we calculated that in this case, about 20% of the phytoplankton, the large phytoplankton flow, carbon flow, was conveyed to the sporangia. And then more than 80% of this flow was conveyed to the zoospore. And so we estimated that, as I said, well, zoospores do not feed, so the loss of this contribution by respiration was minimal. And also, I mean, we calculated maybe only that, for example, the flagella were contributing to the detritus pool, because we consider that while zoospore, when they get eaten, they get eaten entirely. So we realized that this link can be quite effective. So there is a quite, well, not of course 100% of transfer, but finally we calculated that 70% of this flow was conveyed to the, well, the first, the small consumer, because in any case, these models works with the size constraint. So it was logical to consider that we're going mainly to the small consumer and, well, to less extent to the meso zooplankton, but also that the micro zooplankton was contributing then to the diet, to the meso zooplankton. So this link can be direct and indirect, but as I said, we could calculate that was quite effectively. And this then we calculated also for the cyanobacteria bloom. So, well, you can see the carbon biomass that was estimated was larger. It was quite a massive bloom. And again, more than 30% was conveyed very effectively to the consumers. And we also calculated the amount of diet that zoospore can constitute for micro and meso zooplankton. And we calculated that, almost 50% of the diet of consumer can be constituted by zoospores during this blooming habit.
How do you calculate exactly this contribution?
Well, as I say, inverse models are based on mass balance. So we calculated the primary production was calculated. So we had the input flow. And so once you had the input flow, you can calculate all the flows that are going to the different compartments. What we do, we put some constraints. As I say, well, for example, the size constraint was one of the constraints that we used, or you can use, for example, some respiration rate to calculate the loss by respiration. So it's a bit more complex than that, but to simplify, just say that the flow that were not estimated directly were estimated by constraint using the data from the same lakes, because the Lake Paven and the Lake Eda were already quite studied. And when not possible from the same lake, in any case from the literature. So for example, we use it was, I think, about the grazing of the micro and meso zooplankton.
Diet, now we use a size constraint, or you make like the microbiome and then you derive all the states?
Well, it's both. So you make the microbiome, you make some size constraint, and then the flows are calculated. So yeah, and then say these are calculations which are quite realistic, because there was quite some huge work in constraining with the real flow that were estimated, as I say, from the same lake, from the literature, but still these are mathematical models. So is it important at some point when we study ecology to get back, well, at least if not in the real world, to some experiment and try to really measure what happens in the reality. So now we will have a look on how Chytrid supports consumer via the zoospore consumption. Yeah, how this was made by experiment. So we use different models. I mainly use Planktotrix, Maiko, and Ramsey. Hanga, the IGB, used a bit the diatom model, and Utah. So maybe, I don't know if you use a bit more. You use Planktotrix a lot. I don't know if there was something with Asterionella. Alena. Okay, anyway, so just to say that the lab feeding experiments were mainly done with different consumers and few key strains that were isolated and have been our model for quite a while. So the first evidence was that Chytrid supports the consumer grow rate. So this was an experiment that was done by Maiko in 2007. So she used the Daphnia model with the Asterionella, and this ties, you did it here, right? With the Keratella isolated not far away from here.
Across the road, yeah.
So coming back to the importance of having water and small water plant next to your lab. So the first evidence was that consumer can grow when fed with either infected host, and here was only the zoospore. And so, yeah, the same, either with we was used the infected Plantotrix and not only zoospore. But so the following step we have from Thais, I think this was done at the IGB. You were using Daphnia then as a model, was to try to understand where were the process, of course, that were made that, yeah, well, consumers were growing also in this situation. And so it was shown quite nicely, well, that there is an increase of the clearance rate. So, yeah, well, a consumer can feed more. Well, and one of the mechanisms also that has been shown is that actually key treats, well, as we saw it yesterday, in fact, Plantotrix at a very specific point of the cell, and they can also contribute in breaking the filament, so they are smaller. And so this was hypothesized that was also one of the mechanisms that allowed consumers to survive better in presence of infected algae. Another important point that was shown, which is we will see then in a few slides, is more directly linked to the importance of key treats as a nutritional source. It was the reproduction, because consumers, of course, they can feed and grow, but they do not reproduce if they don't have the essential nutrients that they need. So Ramsey was doing an experiment with zoospore and bacteria, and this was quite also strain dependent. So there were some strains that, well, were not reaching the third clutch when feeding only on bacteria. And this is an experiment that we did not so long ago in Lund. So there were Daphnia that feeding on cyanobacteria, they were not reproducing. So they were reproducing only when fed with infected cyanobacteria. So now, as I said, well, reproduction is something that, well, it's really linked to the nutritional quality. So if consumers do not get food of enough quality, they can survive, but they will not reproduce. So what we used to understand what are the underlying process, well, we used the nutritional quality and the fatty acid profile. So I will quickly explain what are fatty acid and how we use them for understanding nutritional quality. So, well, here you have a bit of representation of the three big group of fatty acids: saturated, monounsaturated, and polyunsaturated. So saturated fatty acid, as you can see, well, is just a line. There are no double bonds. They are mainly considered like solid fat, butter, or the coconut butter. So they are mainly considered, yeah, solid fat that is mainly used then for storage. Is stored as a energy, and so this is just saturated fat. As you can see, monounsaturated fatty acid, they have one double bond that start giving a bit more of flexibility to the molecules and a bit more of reactivity also. So these are and even more in polyunsaturated. So you see polyunsaturated have several double bonds. So they are considered very important. For example, as a monounsaturated and polyunsaturated, they are considered very important, for example, as a metabolic fat. So they are not storage fat. They are going, for example, in your membranes, in fat that is used for metabolic process. So, for example, polyunsaturated fatty acid, they are considered very important for the flexibility and permeability of the cellular membrane. So they give this flexibility. They are more reactive, so they are not stored as a fat. So once they are in the organism, they are capable of interact with other molecules. So these, well, they are called metabolic fat, and they are not only stored. And so among the polyunsaturated fatty acids, we have the very famous omega-3 and omega-6 that, well, we know they are quite important also for in the young individual, for what is the cognitive development, the development, well, of brain tissue. So they are actually very important nutritional component. And what I want to add as information here is, well, not all the algae are equal. Not all the algae have the same nutritional quality. So we have a gradient, and this gradient is going, well, pretty much in a, well, good way, and the opposite way then the increase of the temperature. So that's the reason why I was mentioning before also the current scenario, and that global warming, well, is scaring for many reasons, but also because, well, the cyanobacteria, which are, well, bacteria, prokaryotes, so they are known to be rich in bacterial fatty acid, which are known to be highly saturated and even short chain, beside, of course, the presence of toxin, are really, well, known to proliferate in warm environment. As more we go in cold environment, we find algae that are more adapted to this cold environment, and they have a biochemical profile which is considered of higher quality. So green algae, well, and green algae, they are normally used also to feed lab model like, well, the Daphnia, or, well, the Keratella model also was used as a control with feeding on some green algae. So, well, the quality can depend a bit on the general condition of algae, but in general, they are considered a good compromise. They grow quite fast. They are good as a lab food source for consumer model. And when we go in really cold environment, well, the diatom and even more the cryptophyta, we see how the nutritional quality is really increasing. So we have highly unsaturated fatty acid, long chain, so 20 carbon and even more. And so here was, yeah, to show a bit also, as I say, not all the algae have the same nutritional quality. So consumers need also to get the good food source for having the essential dietary component that they need to grow and reproduce. And so this has to show that, well, it's well known since, well, the 90s. There were run some feeding experiment with Daphnia and different algae. And so, well, what they're called deflagellate are mainly the cryptophyte. Here we have cryptomonas, pseudomonas, chromonas. So we see that the growth rate is the highest when the consumer are feeding on this algae. And, well, we have the blue-green algae. Here we have oscillatorial. There we have microcystis. So they really don't grow. And so the information that I want to add here also is that, well, there is a process that is quite known also in food web ecology, which is called the trophic upgrading. So the higher consumers, well, as human as well, they cannot produce what are, well, call it the brick, the short chain fatty acid. So these short chain fatty acid are only produced by algae. And then through the food web, consumers have the capacity, while they are metabolic way and the enzyme that has consumer have, to desaturate and elongate. So add bricks, so add longer chain, and add desaturated bricks through the different steps of the food web. This is the reason why, for example, we get omega-3 from salmon. So they are at the top of the trophic chain. So they had bioaccumulated this fatty acid, but also the things that are on the top of the trophic chain made that they could benefit all the different steps made by the consumer in adding bricks to the fatty acid. So, as I said, this phenomenon is quite known as a trophic upgrading. And so the idea was, okay, but chytrids are consumers, and chytrids are even more complex consumers where we consider the picture of the food web, because as I said, well, they have a very simple life cycle, but still they are consumed as a parasite, they are the zoospore, and then they get fed by consumers. So what are the mechanisms behind that? Can chytrids be trophic upgraders for the food that they consume? And so, yeah, this is the last piece of the work from Michael in 2007. I mean, she was doing really an astonishing work. It was incredible. It was 2007, and she was already realizing that, of course, the nutritional quality of this work can be important. So she measured the nutritional quality of the Asтерionella and the zoospore, and we did it a few years later with the Synedra culture from Silke. And actually, well, as I say, diatoms are known to be of quite good quality. And well, in this case, what we could observe is that the zoospore have basically the same nutritional quality of their host. So what is in the host, chytrids, they absorb it, and it then conveyed via the zoospore to the upper trophic level. Now, what about cyanobacteria? As we said, well, cyanobacteria are not good food quality, and well, the biochemical profile is quite different. So let's start from the monosaturated fatty acid. So what we could observe is that indeed zoospore were richer compared to their host, well, in a bit longer and monosaturated fatty acid. And coming to the polyunsaturated fatty acid, well, zoospores, they were rich in short chain fatty acids, but also they could start having a biochemical profile that was improved and different compared to their host. So we start understanding that, well, chytrids can really act as a trophic upgrader. So there is really an improvement in the nutritional quality that the zoospore can have compared to their host. So it was not simply a mechanistic effect of making the algae more accessible, or allow to feed more on the algae, but there were really some mechanism behind that could make us thinking that, well, chytrids can really have this role as a consumer, as trophic upgraders. And so, well, I'm just going quickly in the last few slides, so I hope you're still with me. So we will have now a look on how to measure really the mechanism of the trophic upgrading with the compound specific stable isotope. So again, just a short introduction about stable isotope. I guess you all know what is a stable isotope is, but just to explain a bit the mechanism that we were looking at. So stable isotopes are variant of a chemical element which different in neutron number. So carbon as a 12, carbon 12, sorry, as a 6 proton and 6 neutron is the most common carbon isotope on Earth. Carbon exists also as a C13, which is the stable isotope of carbon. It exists also as a C14, but at some time this imbalance of protons and neutrons gets unstable. So C14 is a radioactive isotope. Well, it has been used for a while for different measure, and well, fortunately, it's the past. So let's focus on stable isotopes. So as I say, C13 has 6 protons and 7 neutrons. So what is the common way of calling that and say that there is a heavy and a light isotope of carbon. And so here, and in the next slide, I'm taking the cartoon from the Brian Fry book. So again, this is a super nice reference, and while the book is quite, there is quite some knowledge on that book. But yeah, if you have a bit of time, and the cartoons are super nice and are super meaningful. So I just want to mention also this quite important reference. And so, well, isotope, let's say on a chemical point of view, they are quite similar. There is not really a big difference in the chemical reaction. What can be the difference can be in metabolic pathway. So what is known is that sometimes, well, this extra neutron make a difference when there is to climb what it's called the energy hill. So metabolic ways require energy. And so when there is a metabolic way involved, normally the light isotope gets more enriched in the product, because as I say, well, this can be something that, as I said, it can make the difference with the heavy and light isotope. So the isotope ratio can be used to follow the element cycle, and especially to understand what are metabolic processes, or at least if there are metabolic processes involved in the production of certain compounds. So here just a short schematic representation to again guide you a bit through this way. So if we have a consumer fatty acid with the ratio of C13 and C12, well, we have two possibilities. So either this fatty acid has been synthesized de novo, so what is called the enzymatic fractionation, there is some metabolic way involved. So it means that there is some depletion then compared to the diet in the consumer that gets the product. And then we have the dietary uptake, so which is the direct assimilation of the fatty acid as a nutritional source as it is. So there is no metabolism modification involved. So in this first case, as I say, the delta 13, so the C13 amount is higher in the diet compared to the consumer. The consumer is depleted in C13. And in the second case is, well, the opposite, in the sense that we have or either the same signal when there is no bioaccumulation, or sometimes can be a bit higher when the components get accumulated. So this is just an overview of the, let's say, the entire amount of saturated, monounsaturated, and polyunsaturated fatty acid. So as we can see here, for example, zoospore, they get the saturated fatty acid from the algae. And so this means they are accumulated. There is no energy involved. There is no metabolic pathway involved. For the case of the monounsaturated and the polyunsaturated, we see that the signal is lower. So there is a depletion of C13. And so we can imagine that there is some metabolic way involved. And so to conclude, I just want to get into the bit more in detail of two of the short chain fatty acids that we observe. One is the linolenic acid. So when we measure the C13 ratio when in the neonates of the Daphnia in the algae, and in the Daphnia feeding only on Plantotrix. And we can see that the Daphnia feeding on Plantotrix, well, the signal is lower. They need to metabolize this linolenic acid. They don't get it from the food source. So they need it to metabolize. The signal is lower. When we have a look, for example, on a chytrid plantotrix association, the signal is even lower. So there is here some metabolic way involved. So there is some metabolization of this linolenic acid, well, somewhere in the chytrid. So it's not in the algae. And then we can see that the zoospore assimilate from the infected plantotrix. So we can assume that this production of the linolenic acid is happening at the sporangia level, at the infection level. So then it gets accumulated in the zoospore, and the Daphnia have the same signal. So they just assimilated from feeding on the zoospore the same signal. Here, the steradonic acid is more or less the same picture, but we will see what is different in the details. If you remember from the biochemical profile, we could not detect steradonic acid in the plantotrix, while with the higher MS, we could. So this is also a bit the power of stable isotope, of compound-specific stable isotope. It's a method that can be even more sensitive than classical method. So here we could detect a signal, even if the fatty acid was not detecting in the biochemical profile. So the sensitivity is higher. And so we could observe that the Daphnia feeding on the plantotrix, they had exactly the same signal. So they were assimilating from the liddle that were existing already in the plantotrix. When we look at the chytrid plantotrix, we see the signal is almost the same. So there is no metabolic way involved in the sporangia formation. But then at the zoospore, so probably when the zoospore is building their fat storage, here there is some metabolic ways that involved the synthesization of the steradonic acid. And then it gets assimilated. So this was my last slide. I hope, yeah, the story was nice and not too overwhelming, but yeah, just a few key points. So just I hope I could convince you that we were making some work to show how chytrids can be efficient conveyors of nutritional, can be carbon conveyors and efficient nutritional source. They contain essential fatty acids that are very important for consumers, and they can upgrade an inadequate diet coming from different algal hosts that are not as qualitative nutritional as the sport. And finally, yeah, that they can synthesize the polyunsaturated fatty acid that consumers need for their production. So I just want to conclude by acknowledging a certain number of people that was contributing from far or close to this work. I was not doing everything alone, of course. Thomas and Silke that provided the culture that we use as a model, and Kati and Steffi that did a lot of the lab work in Lunds, so a lot of also of this biochemical profile were analyzed thanks to their work. So yeah, this is the end of my story. I hope you enjoyed listening to me, at least a bit, as I did talking.
There's time for some questions.
Very cool.
Yeah.
How long do we take? Okay.
These fatty acids, because so the source host, so the lipid model usually we like to think of these other storage fatty acids separated. Yes, really bad. But is the glyceride or what is the main component? Glycerides? Glycerides?
Yeah, probably, yeah, well,
But then you show that this other SDS
Yeah.
and
yeah, well, yeah.
So these are more like related to, are these more related to membrane fatty acids, or
Well,
is
this-
also part of this storage?
Yeah, well, let's say,
So do you
well,
have an idea about
yeah,
kind of like
yeah.
the
Well,
function?
I would say these are also quite simple organisms. They are unicellular, so I guess that fat that is used as an energy source has this nutritional quality also because, yeah, I mean, it's fat that you can use as nutritional source. The zoospore do not feed on them, but as good quality. So it can be related also to the fluidity of the membrane and, as I said, metabolic fat. In this case, it's not only that. So they use it also as an energy source. I mean, well, they also have some bacterial fatty acids and some unsaturated fatty acids. So they definitely have also some pure storage, let's say, fatty acid. But yeah, they probably use both of them.
You don't know if there is something
No,
about
no.
the function of this.
We
for,
didn't get it.
What kind of or maybe even related to infection process. Because, I mean, we did some, I think that started to also a lot of examples from the plants and parasite interactions where lipids play also a role really in the infection process itself. So I was wondering that.
No, we didn't get into those.
It's a topic to us.
No, but of course it's very interesting. Yeah, it's very interesting. And I don't know if at some point, yeah, I'm sure we will get there to analyze really at the very fine level where is every component of this biochemical profile.
Yeah, I was kind of stricken to see the difference in fatty acid composition between the chytrids that infect algae and the chytrids that infect cyanobacteria. So basically for algae, they have the exact same amylose composition as the algae compared to the chytrids. So that kind of, I mean, means for me that the chytrid in the cyanobacteria has to do some metabolic work to get these things, get these fatty acids. And it kind of is almost a comparison to just the grazers system that they have to, choose between to graze between organisms with different, with better or less good element composition. I think it's an interesting way to look at parasites. That, so in terms of specificity,
if they would have the choice, probably they would have. No, of course, yeah, I see what you mean. I mean, it's also, I mean, at the beginning we were also thinking to run experiment with different consumer at the same time, selective consumer and just unselected grazer to see if, I mean, it is true. If, I mean, it's often consumer that can decide, they know where to go for the nutritional source. I'm not sure if chytrid can do.
This also kind of, like just relating to this because when you think about diversity of parasites of specific host species, I mean, diatoms have a lot.
Yeah, exactly.
Species, but well, cyanobacteria they also and they are more than like one, but still it seems that it's still kind of quite qualitative and not really so quantitative, but it might be also related to.
But you mentioned also some infection mechanism, and so this could be also something. I mean, is it the fact that the organism is missing some essential component that made the infection more targeting some organisms that can provide this? I think we probably get there at some point, but we are not at such a point. But probably also the single cell analysis, we are getting quite precise information with that. So maybe it can be a way. Okay.
Do you ever analyze other types of zoospore, or is it always chytrid zoospores?
These are chytrids.
Yeah, I just wonder if this is a phenomenon specific to chytrids because of the lipid globule, and if we looked at other zoospores that don't have a lipid globule, would we see the same thing? I mean, what we forget sometimes also is that chytrids are fungi, so they are different from other protists, and that's definitely a fact.
Thanks, Serena.
Yeah.
We move on with the next talk. So again, if there are more questions, I'm sure there are. I have here, but we
Yeah, okay.
discuss varieties on the get the floor.